Okay, good morning everyone. Thank you for coming to these 10th anniversary celebrations for the Institute of Developmental Sciences. My name's John Holloway. I'm Associate Dean Research for the Faculty of Medicine. And on behalf of the Dean, Ian Cameron, who unfortunately can't be here this morning, I wanted to welcome you personally to this event. The IDS is a unique strength of the Faculty of Medicine. Since the 1980s, the faculty has pioneered research into the developmental origins of health and disease. Our work has revealed that the environment in which an embryo, fetus, and young child develops influences not only their current health, but their future health and risk of chronic disease. While the concept of the developmental origins of health and disease arose from the pioneering epidemiology of Professor David Barker, and what is now the MRC Life Course Epidemiology Unit, the Faculty of Medicine recognised the need to build on this and connect the epidemiology with molecular biology, cell biology, and developmental physiology to understand the mechanisms underlying the epidemiological observations. This led to the establishment of the Institute to work in partnership with the Life Course Epidemiology Unit and more recently, the Southampton National Institute for Health Research Biomedical Research Centre, to undertake this groundbreaking research. In the decade since its opening, the IDS has shown the power of linking epidemiology, basic and translational science to identify risk factors, understand mechanisms, and develop interventions to prevent the developmental origins of chronic disease. For the faculty, this has led to significant external research funding and enabled us to train a generation of PhD students and early career researchers to take this concept to the wider world, while also contributing to the faculty's core missions of research, education, and enterprise. The fundamental role that the IDS plays in the faculty is reflected in the faculty's mission statement, which is to lead innovation, learning, and discovery across the life course. I want to thank all the staff, students, and supporters, both past and present, who have made this possible in only 10 years. Today is going to be an exciting event. There's lots to look forward to and lots to hear about. I hope you enjoy the day, and thank you very much for being here.
Thank you very much indeed, John. I'm Mark Hanson, and I suppose one housekeeping announcement. We're not expecting a fire practice today, so if the alarms go off, it's for real. So follow the exits and you go out into the car park or along the corridor and out the back. We're here to celebrate 10 years of research, but also, of course, to think about the future. But perhaps we should start a bit with the past. And of course, the idea that development is so important goes back, well, at least 2,500 years, because Hippocrates said that if you have a young growing tree and it becomes damaged in some way, when it becomes a fully grown tree, it may be misshapen and it's not as healthy and happy looking, not as aesthetically pleasing as a tree that had grown perfectly from the beginning. So that's what this wonderful display is all about. This is, folks, the tree of life. And we're starting with little buds and shoots and green things, and you go through, and then when you get to sort of my time in life, it all begins to wither, and it's all very much downward pointing curves. Also, when you get to my age, time goes unbelievably fast, and it doesn't seem like 10 years since we opened the Institute, or actually since 2005, when we laid when Lord Selborne, the Earl of Selborne, came to lay the foundation stone. But actually, to me, it doesn't seem like nearly 18 years since I went to see the Vice-Chancellor of the University of Southampton, Howard Newby, who was a social scientist, along with the then Dean of the faculty, as he is now Sir Eric Thomas, and the then head of the School of Medicine, as he is now Sir Michael Arthur. And we went to see the Vice-Chancellor, and he said, well, what do you want to do, Mark, if you move here? And I said, well, I think, to be honest, we're going to have to try and find the mechanisms underlying David Barker's observations, and we're going to have to try and think of interventions. And he said, so how are we going to do that? And I said, well, to be honest, I think one thing you need to do is to try and bring everybody together, because this kind of blend of science is going to be collaborative. And I think you need to build an institute. Oh, really? He said. And what's that going to cost us? And I said, well, my guess is 6 to 8 million at today's prices. And he looked at me and he said, okay. I said, okay, Vice Chancellor? Yeah, absolutely, let's do it. We went out, and Mick, as he was then, and Eric, as he was then, said to one another, did you hear what I just heard? And we were all just astonished. But that was the vision, and we then moved forward. Of course, it took a long time to finally raise all the funds and start the building. But then it was indeed duly opened almost exactly 10 years ago in 2007. So I hope we're going to have a fantastic day. Our chair, John's welcome to you, especially those of you who have come from overseas, those of you who have fought through the nicely scheduled train strike on Southern. It's called now Southern Railways, isn't it? It used to be Southwest Railways. It used to be Southwest Trains. So obviously they've got a railway, but what they don't have actually is any trains. So let's move on. The first phase then of development starts in fetal life, and the first presentation on developmental physiology and medicine is going to be given by Dr. Lucy Green.
Thanks very much, Mark. What is it that makes you healthy? Why is it that some of us have good health and others, as we approach middle age, suffer from various non-communicable diseases of the cardiovascular system, metabolism, the musculoskeletal systems? We've got to eat better and exercise more, then we'll be okay, right? And in fact, this underpins many government strategies which are encouraging us to eat better, exercise more. And not to mention the ongoing debates about taxing certain foodstuffs. But you know, the impacts are rather disappointing. Obesity rates continue to rise, and it's just really hard. Some of us respond better than others. Some of these initiatives are aimed directly at children in the UK and across the globe. And this is really encouraging to us in the IDS because this is a really key developmental window. But we wonder whether this is just a bit late. That instead that building a healthy baby not only is important to get a viable offspring, but is actually the foundation of lifelong health. It sets you up to be able to respond to the environment in which you're born to. And you know, we're not alone. There is an entire community of researchers working in this field of developmental origins and health and disease. Over 1,000 people met only last month at the International World Congress of this society. This graph shows the membership of the society over the last 10 years and how much it has increased. The graph only starts actually in 2007, funnily enough, when our institute opens its doors. Many of our grouping had actually started to work in this field way before this graph starts, spurred on by the groundbreaking work of Professor Barker and colleagues at the MRC Life Course Epidemiology Unit. So we think that we need to focus very much on those earliest stages of development. Pre-pregnancy, during pregnancy and childhood. And it's the responses and the adaptations and the way they cope will help people survive their pregnancy, but also influence their health as they age. So, how do you build a good baby? Well, building a baby is rather like building a house. You need your bricks, your nutrients, and you need a way of getting those nutrients to the developing fetus. You need a transport system, circulation of the mother, the placenta and the fetus. You need to get all your bricks in order, and for that you need some instructions of how to do it. If you're building a house, that comes from, say, an architect reading the plans, or in the case of the baby, reading the genome. But you know, the fetus can also make adaptations to the nutrients that it receives. And if something goes wrong with those adaptations, or it may alter the baby that is built, just like the house might be altered. And that will influence how that baby copes with the environment that it's born into. It'll alter how that house withstands any hurricanes that it faces. So in our grouping, we're very interested in thinking about how nutrients get to the developing fetus, the adaptations that that fetus can make in response to a change in nutrition, and the lifelong consequences of that. And the things we're talking about here are undernutrition, overnutrition, but also changes in specific micronutrients. So you've got to get those nutrients from the mum to the fetus, and for that you need the placenta. It's so much more than simply a conduit of nutrients from mum to fetus.
Behind every healthy baby, there is a healthy placenta. So you all wouldn't be here if it wasn't for your placenta. So we really should just say thank you to our own placenta. In the Southampton Placenta Lab, we're obviously interested in understanding how the placenta works. So how the placenta works to support the appropriate fetal development. For example, we're interested in how factors in the maternal environment, such as nutrients or toxins, are actually transported across the placenta where they can have effects on the fetus. But we're also interested in how these maternal factors may affect the placenta itself. For example, they can affect placental function or even its molecular makeup. And understanding these placental factors is important because they are actually going to influence fetal development and the subsequent postnatal health. In order to understand these complex relationships between the maternal environment, the placenta, and the placental effects on fetal development, we really need to understand how does the placenta work. We need to understand the basic mechanisms which underlie placental function. And actually, a lot of these factors aren't really known about. So researchers in our group have spent a lot of time researching some of the key nutrient transport systems. For example, we have shown for the first time how the key nutrient amino acids are actually transported from the maternal circulation across the placenta and into the fetal circulation. We've shown the actual mechanisms which allow amino acids to be released into the fetal circulation, and we've identified new transporters on this fetal side of the placenta. So these are really key mechanisms which are going to have an influence on fetal development. But as you can see, as for most nutrient transport systems, this is a complex process with many components. So we need to start understanding how these systems work as a whole, rather than just focusing on individual components alone. To do this, we've been working with collaborators in engineering, where we've put together mathematical models of each of the nutrient transport systems. So what we can do is we can model some of the nutrient transport systems, such as amino acid transport. We can depict, as shown down here, how, say, amino acid transport will change by changing each of the components of the model. So, for example, those fetal-facing amino acid transporters I just mentioned. And we can see how all of these factors sort of work as a whole to influence the overall transport. And we've had some interesting findings because we actually see that metabolism has a key effect on the transfer of amino acids across the placenta. We also see this for our models of lipid transport, but it's not important in our models of cortisol transport. So we're working with our functional studies and our modelling studies to actually see how the placenta works. But we also need to take into account the placental structure. So new 3D imaging techniques have allowed us to actually take our functional studies, where we're looking at, say, the whole placental level at the individual cotyledons, right down to the villus tree structure, and even down to nanometer scales, where we can look at the structure of the microvilli on the maternal face of the placenta. Our work is focusing on all of these levels to look at the interactions between placental function and structure and the effects of the maternal environment and how the placenta influences subsequent fetal development. So our mechanistic studies have identified lots of key components in the placenta that could be important for fetal development. So to test this, we've utilised placentas from some of the Southampton cohort studies, including the Southampton Women's Survey, where we've looked at measures within the human placenta and related them to both fetal and neonatal outcome. And one such example is shown here, which is these amino acid transporters on the fetal side of the placenta. We've actually measured these and related them to fetal outcome, and we see there's a positive relationship between this amino acid transporter and lean mass at birth and at 4 years of age. So this is supporting our idea that the placenta is really important for supporting fetal development. We want to now know what is actually regulating these mechanisms within the human placenta. So again, we utilise data from the Southampton Women's Survey, and we can look at factors in the maternal environment and relate these to the mechanistic studies in the placenta. And one such example is shown here, where we actually find the maternal arm muscle area relates to the activity of one of these key amino acid transporters in the placenta. Our studies have also shown relationships between other maternal factors such as diet, such as exercise, and also factors such as smoking actually relate to components in the placenta. But one factor I'm particularly interested in at the moment is maternal vitamin D status, as studies in this Southampton Women's Survey have shown that maternal vitamin D levels relate to the expression of a number of these key amino acid transporters. We all know that vitamin D is important for appropriate fetal development, but we don't actually know how vitamin D, so the inactive vitamin D or even the active vitamin D, are transported across the placenta. It was thought to be simple diffusion. We don't know how vitamin D, which is a key transcription factor, actually affects the placenta itself. So there are a lot of unknowns when it comes to the placenta. But our work has now shown how vitamin D is taken up into the placenta, and this is a regulated process. So this graph shows that vitamin D uptake actually occurs in both a time and a temperature-dependent manner. So at 4 degrees we see no uptake, and at 37 degrees, shown in green, we see uptake of vitamin D into the placenta. So this shows that it's an active process, and further experiments with specific blockers have shown that this is actually via endocytosis. So we've discovered novel mechanisms of nutrient uptake into the placenta. We've also shown, again using human placentas and studies in the sheep placenta that you will hear about later, that vitamin D is actually metabolized by the placenta itself as it crosses. So the placenta not only controls the amount of vitamin D taken up, but it also controls the amount and the type of vitamin D being released into the fetal circulation to affect fetal growth. So if we're getting metabolism of vitamin D, we're getting an active vitamin D product, and this is the product that has effects on gene transcription, so it can be having effects on the placenta. To investigate this further, we again looked at human placental fragments, and we looked at how vitamin D would actually influence the makeup of this placental tissue in terms of gene expression. Our data shows that vitamin D, when put upon placental fragment samples, has key effects on this tissue. It affects DNA methylation, so these on-off switches for gene expression. It affects gene expression, and it actually affects the functional proteins. So vitamin D is upregulating a number of key pathways in the human placenta, as well as downregulating a number of pathways. And an integrated analysis of all of this data is showing that vitamin D is upregulating the chromatin remodeling of the placental tissue. So what it's doing is it's actually activating the placenta at a cellular level to switch on genes to carry out growth-promoting effects. we're also seeing that it's downregulating the inflammatory pathways, so it's perhaps having a protective effect in the human placenta. So if it's having these effects on the placenta at the cellular level, it's going to be affecting placental function. And this may be mediating some of the effects we see when we relate maternal vitamin D status to factors in terms of fetal growth and subsequent postnatal effects. So again, we move back to our cohort studies and look at the Southampton Women's Survey placentas. And what we've done is we've looked at some of these components in the vitamin D system in the placenta and related them to fetal and neonatal aspects. And for example, we see that the vitamin D receptor, so one key component in the response to vitamin D, does relate to weight and lean mass at birth and at 4 years of age in the offspring. So we're seeing that maternal vitamin D status relates to placental factors and to fetal outcome. So this perhaps provides a mechanistic basis for some of our intervention studies. So our work has moved into the Metabolism study, which is a randomized control study where women have been supplemented with vitamin D during pregnancy. We've collected the placentas from this cohort, and we're again looking at the molecular makeup of these placentas to see how they're regulated by the vitamin D supplementation during pregnancy. We're again looking at DNA methylation, gene expression, and protein expression. And what we can then do is relate these factors to both fetal and neonatal growth in this cohort. So the work so far in the Southampton Placenta Lab has actually established some of the key mechanisms in terms of placental function, and we're now beginning to show how these are regulated by the maternal environment and how they have key effects on fetal development as well as subsequent neonatal development and lifelong health.
So what can the fetus do if it, in response to changes in the nutrient that it does receive? One of the strategies it can take that can be taken is that gestation length is altered. We and others have shown that milder challenges of undernutrition during pregnancy can cause gestation length to be slightly longer, a little bit more time for that fetus to develop. Others have shown that a more severe undernutrition challenge can actually lead to a shorter gestation. Perhaps it's actually just better to get out while you can. But there are other ways that the fetus can adapt. It can change where it sends its nutrients and what is done with those nutrients. Work in the Southampton Women's Survey has found that fetuses can redistribute where they send their blood flow in response to the mum being thin or eating an imprudent diet. Actually, more blood flow goes to the liver. And in sheep, we investigated this a little deeper by specifically manipulating the amount of glucose that got to the fetus and found that the fetus does make a coordinated cardiovascular response with less blood flow going to the adrenal, more going to the adrenal, less to the liver, and less through the femoral arteries into the hind limb. And we found that actually this femoral artery blood flow response, the speed and size of it does depend on the nutrition that the mum got earlier on in pregnancy. So even during fetal life, the way that the fetus responds is influenced by what went on before. And we've carried on this work to look specifically at vitamin D deficiency and the kind of responses that the fetus makes to that. First of all, we found that even an undernutrition, just total calorific restriction to the mum in early gestation, changed the amount of vitamin D that the fetus had in late pregnancy. It actually had less of it. And we found that this is due to changes in the way that the placenta handles the vitamin D, rather like what Jane was telling you about a minute ago. We went on in collaboration with a number of investigators across our institute and with people from the MRC Life Course Epidemiology unit to set up a sheep pilot study where we specifically lowered the amount of vitamin D in the fetus and found that these fetuses, in response, had a thickening of their heart walls, shown here. Their blood vessels in their legs didn't relax as well, and their bones in their hind limbs actually had a weaker structure. Our challenge now is to really align these sorts of data with ongoing trials carried out in Southampton, some of which actually supplement vitamin D during pregnancy. And we can learn a lot from those data, but also potentially add some mechanistic insight. This sort of adaptation might well have some impact on these individuals as they get into postnatal life. And we're chasing up some of these ideas in terms of skeletal muscle function in postnatal life, and there's a poster on that outside. But what about the long-term consequences? We carried out some work a few years ago which showed that actually prenatal undernutrition to the mum, that's this bar here, caused in the adult offspring there to be a thickening of the heart walls. And also the way in which their coronary vessels constricted was affected. What was really interesting from these data, though, that if you minimise the mismatch in undernutrition between the prenatal and the postnatal environment, those effects were minimised. And really what this said to us is that it's really important to keep an eye on the balance of nutrition between prenatal and postnatal life. And actually, when you don't, that's when the biggest problems may come about. And that these problems may well cross generations. This data in rats from our investigators showed that protein restriction during pregnancy does impair cardiovascular function in the grandchildren without any further challenges taking place. So we're talking here about them having a higher blood pressure, impaired relaxation of their blood vessels, and impaired marks left on their on key genes that regulate cardiovascular system. I've told you about the fact that in response to undernutrition, often the fetus is sending blood flow away from its extremities, away from its limbs. And so we found that actually that can impact on how those limbs grow. The muscle in them grows and develops. And we've measured this using histology techniques to count myofibers and found that undernutrition in the mum during gestation leads to less fiber development in the fetuses during fetal life. And that these offspring may go on to not be able to handle glucose as well when they get to adult life. And even if, as they get older, they can put this right and handle glucose well again, they still show a lower density of myofibers and capillaries in their hind limb muscles. So long-term effects. Don't get us wrong, we've not dismissed childhood as an important developmental window. Indeed, some of our sheep work has shown that actually if you undernourish a sheep just after it's weaned from its mum, it slows its growth rate. And when it grows up, we can pick up the fact that it has an improved glucose tolerance, perhaps maybe in order to right its growth. And this is due to an improved insulin sensitivity. But that comes at a cost because actually these adults' offspring lay down more fat. It's a girl thing, this, and it may be something bound up with reproductive strategy. Of course, I've talked to you about undernutrition. In fact, one of the big problems in Western societies is overnutrition for women, men and children. 1 in 5 women in the UK are obese, and this is going to impact on them during pregnancy too, with impact on their health, but also their offspring's health. Our researchers in mice models have fed a high-fat diet to cause obesity in the mum during pregnancy and have found that their offspring don't handle glucose as well, rather like in a person with diabetes. And these offspring themselves are fat and have laid down a lot of fat in their livers. And our researchers have shown that this is due to an impaired function of the enzymes in the mitochondrial electron transport chain, which means they don't handle fatty acids as well, and they tend to towards pathways that lay down more fat. The offspring of these these mice who are obese during pregnancy also have a higher blood pressure, and their blood vessels do not work as well, do not relax as well. The vessels show increased levels of oxidative stress, as do the skeletal muscles that they supply. So really what we're seeing here is that offspring of a high-fat, obesogenic pregnancy have a cardiovascular and metabolic dysfunctions. So what do we do about this? Is it all about managing weight pre-pregnancy and during gestation? Actually, women who are obese with metabolic problems can be given drugs during pregnancy. Things like metformin, the anti-diabetic drug, or statins, the cholesterol-lowering drugs. And sometimes this is an attractive idea because it may be better and more compliance and more efficacious. But what is the impact on their offspring? Well, our investigators have tried to work this out. Our researchers have given metformin treatment to obese pregnant mice and found that actually less fat is laid down in their adult offspring's liver. And it's working to protect against the dysfunction in that electron transport chain so that less fat is laid down. Our researchers have also tried to give statin treatment to pregnant mice who are on that high-fat diet and found that it actually reduces cardiovascular risk factors in their offspring. So their blood pressure is no longer as high and neither is their cholesterol. It's a slightly different approach. Our researchers have also given folic acid supplementation in an attempt to put right some of the dysfunction during decreased energy during gestation. In this case, because of a protein restriction in the diet during pregnancy. And have found that actually folic acid supplementation can put right the dysfunction in the maternal uterine vascular function, but also it can restore function in their offspring's blood vessels too. So we've produced some evidence really that a number of different sorts of nutrient challenges across pregnancy can cause the fetus to make adaptations and with lifelong consequences. We're doing what we can to put a stop and intervene in this type of pathways. The sorts of challenges that have an effect can be at various different time windows during pregnancy, sometimes really early on, even before pregnancy. And this is important when you think about the Dutch winter famine and the early gestation exposure to the famine that had lifelong cardiovascular consequences. But how early is early, and how is it all working?
So, how early is early? How important is the environment during the peri-conceptional period? That's the time when we are usually not aware yet that we are pregnant. So, with a long-standing collaborative effort, together with biological sciences and clinical experimental sciences, we have investigated this in a mouse model using a low-protein diet fed to mothers only and exclusively for the first 3.5 to 4 days of development. That comprises the pre-implantation period only. And what we found in the offspring is that this very short challenge changes how the body works for life. And specifically what we found in the offspring is that they experience trouble in cardiometabolic functions such as increased weight and increased blood pressure. They even change their behavior. They become more active, but also more forgetful. And all these phenotypes, they persist into adulthood, even in the presence of a thereafter normal diet. We now know as well that all these changes in adulthood are induced by the time the blastocyst develops, which is as early as 3.5 days. And this is a critical stage in development at which time you actually undergo the very first differentiation into more specific cell phenotypes in life. So how does the blastocyst actually manage to adapt to these challenges in the environment? And we find in this low-protein diet model, we find that the maternal circulation here is low in certain amino acids and insulin, which is also translated into amino acid levels within the womb, which are lowered. This is the environment actually where the embryo develops in. And if we are now looking at the blastocyst a bit more closely, we see a whole cascade of compensatory mechanisms being triggered at that particular stage. For example, the outer cells, these ones here, which are responsible of taking in the nutrients at that point and are also going to develop into the placenta later on, they grow more. Certain signaling pathways within the blastocyst, which are involved in sensing the nutritional environment and nutrient availability, and also regulate thereafter the growth and differentiation processes accordingly, they are altered. The blastocyst increases its nutrient intake via endocytotic mechanisms, and to make use of those extra nutrients, to build more building blocks of life, of cells, such as, for example, proteins, it also upregulates the machinery that's necessary for these processes, such as ribosome biogenesis, as well as other mechanisms related to protein synthesis. It looks highly likely that epigenetic regulation is involved in all these processes. So we now know that the blastocyst has got a certain toolbox it can utilize to adapt to its prevailing conditions in the environment in an undernutrition model. And how about an overnutrition model? More recently, we've been focusing on challenging mouse mothers with a high-fat diet, again exclusively during the pre-implantation period, so for a very brief period, 3.5 days in total. This is insufficient time for the mothers to actually become obese, but what we do see in the mothers is that they are less capable of handling glucose normally during that particular time. And what we see again is altered blastocyst growth, this time affecting the entire blastocyst and consistent with a more advanced differentiation of that particular blastocyst. This is matched by a earlier preparation for pregnancy of the uterus. Metabolically, we see as well in the blastocyst that it's not as capable to handle glucose normally as compared to the controls. There's a poster outside which goes into this in a bit more detail and also shows the cascade of mechanisms that are induced by this particular feature. So what happens after normal diet is resumed? Again, 3.5 days dietary challenge followed by a normal diet thereafter. And we see indeed the changes we see in the blastocyst, they do persist. We see, for example, the impaired glucose handling capability is continued within the fetus, even though the normal diet has been resumed for about 14 days. We also see changes in how the fetus and the placenta grow. We can measure these, and we see that both change their relative weights. We also have observed that the bone mineralization in response to a high-fat diet for 3.5 days is advanced and increased. Within the offspring, we again then see difficulties in cardiometabolic features such as impaired glucose handling early on in adulthood, followed by a little bit later increased blood pressure. These animals again change their activity levels as well, and this coincides with a change in brain structure, for example, an increase in microglia, which may suggest a pro-inflammatory state. So in short, we've now seen that the blastocyst is capable of sensing and detecting its environment and adapting its developmental processes accordingly. And this may tell us that we have to start and look after our embryos much earlier before we even know that we are pregnant. All this knowledge can be useful as well for, and has implications for infertility treatments where embryos are either manipulated and taken out of the normal environment and exposed to in vitro culture conditions. The knowledge we have may help us improving these conditions and minimize long-term effects. In fact, we know now that how what we eat affects the environment we experience very early on in the womb in much more detail. So, for example, here our researchers have shown that certain amino acids are actually upregulated within the womb in response to a negative diet, a negative prudent diet. If you eat more healthily, these certain amino acids are at a normal level. This knowledge can help us to develop ideas what the optimal environment is for a human embryo. With all this in mind, we've set up a clinical trial, a randomized clinical trial, in which we investigate how changing what the mother eats prior to undergoing fertility treatment may affect the outcomes. This is called the PREPARE trial and is currently running here in Southampton. The mothers are given a Mediterranean diet and are mainly, which is supplemented with vitamin D and fatty acids, omega-3 fatty acids, for 6 weeks prior to conception. And the researchers will be looking at how the embryos develop and how the uterine environment will change in response to such a diet. Is there an easy way how to measure this uterine environment? And indeed there is. Very pioneering and groundbreaking work is being pursued here to investigate exactly that. And the researchers are developing a microsensor device which is designed to monitor the uterine environment in real time, currently concentrating on oxygen tension and pH. And this little
The ability to be able to monitor the womb's environment will mean that doctors can select the right patient for the right treatments. Our sensor device is invented just to do that. It is a miniaturized sensor that the doctor in the fertility clinic can insert into the womb. It is batteryless, wireless, and is powered by a belt that the patient wears. The data is transmitted to a mobile phone and will be analyzed through a diagnostic algorithm to inform the doctor of the right treatment for the patient.
So overall, we now understand that even the very early period around conception, what we experience there matters for our entire lives. We are beginning to understand the mechanisms, how these changes are brought about, and we are also developing technologies how these changes can be measured in the human.
So our work over the last 10 years has really reinforced to us that we shouldn't be waiting until we start crumbling in middle age, or perhaps indeed until childhood before we try to put things right. We have a really prime window of opportunity where the maternal environment is having an effect on an individual. But you know, this has got to be a really two-way conversation between human trials and the animal models, where the human trials are using novel technologies and trials to really understand how to optimize the pregnancy environment. And the animal models are really working out some of the core mechanisms and ways to actually intervene and put things right. But, you know, we have to be really cautious about these sorts of interventions. Work going on in our institute is showing that, for example, antiretroviral treatment of women who have HIV during pregnancy is of extreme importance and benefit, but may have unintended consequences for the babies who are at risk of being small for gestational age or at preterm delivery. But, you know, overall, if we can continue to work with our colleagues across the university and within the NHS, we've got a real opportunity to make a big difference and to develop ways forward in which we can help inform health professionals, from midwives to doctors to nurses, and also future parents. For us, it's all about promoting health across the generations. Only a few of us have stood up here today to talk to you about our data, but done this on behalf of all of these principal investigators listed here. Of course, none of this work would have got anywhere in the last 10 years without the tireless and often superhuman efforts of the staff and students that have made up these groups in that period of time. And we thank you all. We also thank all our collaborators, past and present, who have enthused and stimulated and just rolled up their sleeves and got on with their work to make all of this happen. And to the bodies who have funded our work. Thank you very much.
Thank you very much, Lucy, Jane and Judith. You've really opened up the black box for us, really, of fetal life, embryonic life, and shown just how much is going on there and what the opportunities are. I was just thinking, when I started out, became interested in development in probably the early 1980s, it was partly triggered by hearing a very eminent physiologist, talking to a friend of his, I think at the bar, saying, yeah, well, of course, to work on development, you don't have to be that clever. And I'm sure he was referring to me. But it made me think, is that really true? The attitude then was, well, real physiology, real science starts later in life. You know, we run on a clockwork kind of thing, you know, that's all programmed and it's all set up and you can't change it very much until the child is born, and then, of course, real life starts. And that's totally wrong, not only from the observations of David Barker and others that, of course, that period of life sets up risk, but also now all the science that we're hearing about that shows that the fetus can respond, the placenta, which, of course, is usually just thrown away, can respond in all sorts of ways. Even the embryo is sensing its environment, and actually the mum and dad are setting up the story even before it begins. So thank you very much for that introduction. I think, if you'll forgive me, folks, we'll save questions until the end because I'm sure we don't want to run too much into the lunch break. So hold those burning questions and we'll try and get some time at the end for discussion. So we've heard quite a lot about nutrition, haven't we? But really what nutrition means is very different depending on who you talk to. It's not the same as food. It's not necessarily the same as diet. And anyway, nutrition must, to a certain extent, depend on metabolism. So our next speaker is Professor Philip Calder, who's our Professor of Human Nutrition, who's going to take up that challenge and tell you about the work of that wonderful group over the last decade. Philip.
Yeah, great. Great, thanks very much. So I think Lucy, Jane and Judith have done a fantastic job with that presentation. That was really amazing. And they've done me a great service, I think, because they've really shown us quite clearly the importance of early life nutrition, of understanding metabolism at the earliest stages of life, and what that means for the embryo, the fetus, the mother, and the baby once born, and what the lifetime consequences of some of those differences might be. That enables me to deal a little bit more with some of the work that we've been doing at the other still important stages of the life course, which is good. So there's two little bits of overlap between the two presentations because quite wrongly we didn't talk very well to one another about it, but I don't think that matters too much. So it's a huge pleasure for me to be talking to you on behalf of the PIs in the Nutrition and Metabolism research theme. Those PIs are listed here. We also, as you will have heard already, we work very closely with other colleagues within the IDS on their projects related to nutrition and metabolism, and with people in the MRC unit and within the Biomedical Research Centre. So what we're really interested in is the influence of differences in food or nutrient intake on nutrient status, which of course involves handling and metabolism, and then the impact of nutrient exposures on growth, development, behavior, physiological and psychological function, on optimal ability to respond to challenges. Some people call that resilience or fitness, and therefore the influence of nutrition on health and remaining healthy as we age, on disease risk, and then on disease progression if we develop a particular condition. Of course, our diet changes during the life course quite a lot, and part of that is driven by our physiology and our requirements, but there are also other influences on our dietary intake and therefore our nutrient intake. Some of these can be cultural or social, but there are also psychological influences, and of course disease has a major impact on our food and nutrient intake. So there's a changing pattern of dietary intake, of nutrient intake, and therefore of nutrient status across the life course. What I'm going to talk about is our work across the life course. I'm going to show you some highlights from the last 10 years with regard to our research. There's a little bit on what we do in education. On the way, you'll see some of our impacts, a little bit about public and patient engagement, and I think there's a slide on enterprise. And then I'll finish up by telling you something about the future. I think one of the strengths which we have, which actually the others already alluded to, is a translational approach, where we use in vitro or animal systems to generate new discoveries and to understand mechanisms, and then we look at the outcome of that information either through epidemiological investigations in cohorts or through intervention studies. And I'm going to talk quite a lot about some of the intervention studies that we've done, although not in a lot of detail. Many of these, of course, are done in free-living, relatively healthy or pre-disease individuals, but also we have interest in many different patient groups. So you've already heard a little bit about this experiment, which is an animal feeding experiment where pregnant mice received a normal low-fat diet or a high-fat diet during pregnancy and during lactation. And then after birth, the offspring were switched amongst different types of diets, so that ultimately 4 different groups were generated, determined by the pregnancy and lactation exposure and the post-weaning exposures. So what was seen was differences in body weight, differences in glucose tolerance, therefore insulin sensitivity and glucose metabolism, and big differences in liver fat. So the maternal nutritional environment has an impact, but there's also an interaction between the maternal nutrition environment and the offspring nutritional environment to determine the ultimate metabolic and body compositional phenotype. The interesting thing in this study is that the offspring also showed big differences in physical activity, as you'll see here, looking at physical activity over time in the 3 different groups, in food intake, and in expression of genes involved in controlling circadian rhythms. So it's fascinating that the entire physiology and behavior of these animals is influenced by these dietary changes early in life. There's also an impact of these early life exposures on the microvascular density in skeletal muscle, and therefore in the supply of oxygen and nutrients to skeletal muscle, and of course this will have an impact on body composition, on activity, and on metabolic capacity. As well as a translational approach, of course we're very interested in the life course, which is why we're here today. So what I'm going to do is talk about some of our interventions at different stages of the life course and show you some of the data. So Judith mentioned the PREPARE study, which is an intervention in couples awaiting in vitro fertilization, where both the man and the lady of the couple are randomized to either the Mediterranean diet group or a control group for 6 weeks until the egg and sperm are collected and then the IVF happens. And the principal outcome of this study is embryo quality. And what we have found is the quality of the embryo is better in the group consuming the Mediterranean diet compared to the control group, although pregnancy success rates are not different between the two groups. So it'll be very interesting to follow up the offspring from that study. Moving into pregnancy, we've conducted really a landmark study, I think, called the Salmon and Pregnancy Study, which was a dietary intervention with salmon in women who were self-reported low oily fish consumers. The intervention lasted from week 19 of pregnancy until they gave birth. If they weren't in the intervention group, they remained in the control group, continuing on their habitual diet. We took numerous samples from the ladies during pregnancy, cord blood, cord tissue, placenta, and so on, breast milk up to day 28. And we followed the infants at 6 months and 2.5 to 3 years, mainly for outcomes to do with allergy and asthma, because that was the hypothesis that we were testing, that early exposure to oily fish would reduce the risk. So one of the, I think, really important findings that we made in the Salmon and Pregnancy study is that the vital omega-3 fatty acid, docosahexaenoic acid, which is delivered to the mothers through oily fish during pregnancy, is higher in the breast milk of those women going right out to 1 month after giving birth compared to the level in the control group. And DHA is very important in infant visual and brain development, in development of the vasculature and the immune system. So women eating oily fish during pregnancy are better able to deliver this very important fatty acid to their breastfed infants at least out to 1 month after birth, even though the women stopped eating the fish at the time of birth. So we have some new data which we haven't published from the follow-up of these infants when they reach 2.5 to 3 years of age. So these are the proportions of children in either the control group or the salmon group who were reported to have wheeze in the last 12 months or to have doctor-diagnosed asthma. And you see those proportions are much lower in the offspring who receive salmon, whose mothers receive salmon during pregnancy compared to the control group. So I think this is going to be a very important finding that we've made. Moving into childhood and adolescence, we've been involved in quite a number of different interventions, mainly with omega-3 fatty acids, because several people in the group are interested in omega-3 fatty acids because of their bioactivity in relation to inflammation, immunity, and cardiovascular health. So this is the results of a study that we did in Thai school children, who for 6 months were involved in a randomized controlled trial, and they consumed a milk-based drink which contained vegetable oil as placebo or omega-3 fatty acids as the active group. During the course of the 6-month intervention, they had nurses going into the schools and monitoring the outcomes in the children to do with illness. So in the group who received the omega-3 fatty acids, a lower proportion of the children over the 6 months had illness with fever. There were a fewer number of episodes of illness per child, and the number of ill days when they were ill was significantly lower in the omega-3 group. So it seems improving omega-3 fatty acid supply to these children has a benefit in terms of illness, and this was actually mainly respiratory illness in that group. More recently, in a study in collaboration with researchers in South Africa, we have done a trial comparing iron alone, omega-3 fatty acids alone, or the combination of iron plus omega-3 fatty acids versus placebo. So these were iron-deficient school children who were given the supplements of either iron or omega-3 fatty acids for about 180 days. And this is the occurrence of respiratory symptoms over the course of the study. So this is the placebo group here in the dark line. So around about 30% of children in the placebo group developed respiratory symptoms over the course of the study. Giving them iron, even though they were iron deficient, made the situation worse, so that about 70% of them had respiratory illness during the course of the study in the iron group. If they were also given omega-3 fatty acids, the omega-3 fatty acids acted to mitigate the deleterious effect of iron. So there was an iron omega-3 fatty acid interaction, although omega-3 fatty acids on their own had a very small effect in this particular study. So I think this is very interesting in terms of nutrient interactions to determine a clinical outcome. There's been some interest in adding bioactive fatty acids to ready-to-use therapeutic foods that are used to treat children with severe acute malnutrition. So this is results of a study that I was involved in, conducted in Malawi, simply showing that you can deliver more of these bioactive fatty acids, eicosapentaenoic acid or EPA, and docosahexaenoic acid or DHA, through this route of therapeutic foods in these children. There was a difference between the delivery of the fatty acids depending upon the starting status of the fatty acids, which makes, I think, some biological sense. So if the children started with low status, they actually had higher incorporation of the fatty acids into blood lipids, and into red cells and white cells. And there was a small functional benefit of the omega-3 intervention in this trial. Our colleague Richard Holt is very interested in factors that influence the ability of adolescents to control their diabetes. So I think this is a very vulnerable period for an individual to find that they have a disease like diabetes and what the long-term consequences of that might mean to them. So of course these individuals show the pathophysiological changes, but they also, along with the disease, show psychological changes and sociological changes. And what Richard's work has shown is that understanding all of these different factors is very important in determining how well an individual will follow the correct path to controlling their condition. And if they don't control the condition, of course, it gets worse. So there's an elevation in blood glucose, or in uncontrolled episodes of blood glucose, and hemoglobin A1C, and more frequent diabetic ketoacidosis admissions. So understanding the factors that help these individuals control their diabetes is very important. So Richard has published a series of papers on the psychosocial barriers to people managing their own diabetes. And I think these are very fascinating papers showing that one must consider the psychological and social consequences of having a disease if you want to understand the ability of individuals to self-manage the disease. Of course, our research also very much impacts our teaching. So we have an MSc course in diabetes, and we have this fascinating MOOC on understanding insulin that Nicola is going to tell us about now.
Teaching in the Institute of Developmental Sciences comes in all shapes and sizes. Many of us teach undergraduate medical students, master's students, PhD students, or actually other groups of people as well. Most of our teaching occurs face-to-face, which gives a great student experience, but we wondered if we could reach an audience who couldn't actually make it to Southampton. So we developed a MOOC, which is a massive open online course, which students can access for free from their online terminals. This MOOC educates about insulin by following the story of two people who have type 1 or type 2 diabetes. And our aim is really to make this education reduce the errors made in prescribing insulin to people with diabetes. The MOOC is really interactive. We have comic strip style patient scenarios, we have Twitter feed animations, we have quizzes, diagrams, videos, as well as a discussion forum. The first time we ran the course earlier this year, we had about 1,600 learners, which was really amazing for us. And they came from more than 70 different countries. We realised that quite a few of those countries were in areas of the world that would find it difficult to send people to attend traditional courses in the UK.
Thank you, Nicola. You could have come up and done it live, but great. So moving into adulthood, of course, this has been a great focus of our research for many years now. And as you'll be aware already, we're very interested in omega-3 fatty acids. And this is an example of a study we've done in healthy volunteers, trying to identify how the status of the bioactive omega-3 fatty acids, EPA and DHA, changes when people increase their intake of EPA and DHA, and whether that's different if you look in different places. So the thinking here is, of course, we digest and absorb our fatty acids. They appear in the bloodstream where they're transported as components of different lipoproteins, and they may have different fates. Some of them may go to the liver to be further metabolized. Some of them may go to the adipose tissue to be stored, and some of them may go to different functional cells and tissues where they play a role in modifying cell membrane composition and influencing the function of the cell membrane in the cell itself. So in this particular study, we asked the question of whether different places in the body, in the human body, change their EPA and DHA status in different ways according to the increased intake of those fatty acids. So we did a dose-finding study over 1 year in 210 healthy volunteers who were divided into 5 groups in the study. Here, data for 4 groups is shown. They were either in a placebo group or in a group receiving 1 of 3 different doses of omega-3 fatty acids. This simply shows the appearance of DHA in one of the plasma lipid pools as a function of time in those different groups. So you see there's a very nice dose-response relationship, and there's a particular time course. Now, the details of this study don't matter to you today, but what the study identified was the nature of the dose response and the time course incorporation of these 2 bioactive omega-3 fatty acids into 9 different pools in the human body over a 12-month period, identifying very novel information about how our body changes upon exposure to these bioactive fatty acids. Another one of our interests is in prebiotics, probiotics in the gut microbiota, and how modifying the gut microbiota might impact on our immune system. Because the thinking is that dysbiosis, that is a non-optimal gut microbiota, although that's very hard to define, results in an impairment in host immune function that maybe has both metabolic effects but also immunological consequences. So we've conducted some randomized controlled trials. This is with a common prebiotic, the beta-2,1 fructans from inulin, and using seasonal influenza vaccination as the readout for the functionality of the immune system. So the key finding of the study is that the increase in antibody titers to one of the 3 strains of the seasonal flu vaccine can be enhanced if people are given a period of about a 4-week supplementation with prebiotics compared to the control group. So this is the antibody titers here over 4 weeks post-vaccination, showing the enhancement seen in the group receiving the prebiotic, and of course this was linked to alterations in the gut microbiota, particularly an increase in bifidobacteria, which other people have shown to be very important in determining anti-influenza vaccine responses. Moving into how did you describe it, Mark? What was happening at the end of life? We were withering or wilting or everything was drooping or something like that. So work in older adults and adults at risk. So one of the areas that we're very interested in, led by our colleague Christopher Byrne, is non-alcoholic fatty liver disease. So this is a very prevalent condition. Probably 30% of adults in the UK have non-alcoholic fatty liver disease. The prevalence in obese people may be as much as 80%. This is a condition which is clearly driven by obesity, but involves fat deposition in the liver, which is not a place where fat should be deposited. And in combination of this altered lipid metabolism with oxidative stress or inflammation, the condition can be driven to a more serious condition, ultimately leading to liver cirrhosis or liver cancer. And it's clear that altered metabolism and perhaps altered nutrient exposures in association with some other factors like particular genotypes are very important in determining the condition and its severity and its progression. So having a fatty liver itself is not a good thing because of the increased risk of liver cancer, liver cirrhosis, and requirement for liver transplantation. But also, having non-alcoholic fatty liver disease increases the risk of many other conditions, particularly cardiovascular disease, but also renal failure and so on. So it's a very important condition to understand more and to design interventions to slow, reverse, or treat. So we think one of those interventions could be omega-3 fatty acids because these influence hepatic lipid metabolism, they influence inflammation in the liver and in adipose tissue, and of course they also influence lipolysis in adipose tissue, reducing the flow of fatty acids to the liver, which are the substrate for the triglyceride which is deposited. So we conducted a landmark study called the Welcome Study, the main results from which were published in Hepatology in 2014. This is a randomized controlled trial of high-dose omega-3 fatty acids in patients with non-alcoholic fatty liver disease. 100 patients randomized to one of 2 groups, placebo or omega-3 fatty acids. The study was very long, 18 months duration. So this is the highest dose and longest duration omega-3 fatty acid study done in NAFLD. We had extremely high retention with 92% of patients finishing and extremely good compliance. So the key finding from the study was related to liver fat, of course, and what we identified was that liver fat loss was better in the omega-3 fatty acid group than the control group, and using multivariate analysis, despite the randomized controlled trial design, to identify predictors of liver fat loss, we identified 3 factors that were really important. One of them was loss of body weight. So if you lost more body weight, you showed a greater reduction in liver fat. The second was starting liver fat. So the greater the amount of liver fat you started with, the higher was the loss. And the third was an increase in omega-3 fatty acid content measured in red blood cells as an indicator of tissue levels, showing the higher the omega-3 fatty acid or DHA incorporation, the greater was the liver fat loss. And omega-3 fatty acid incorporation into red cells was an independent predictor of liver fat loss, perhaps working through those mechanisms that I talked about before. So our study was very influential on the recent NICE guidelines around NAFLD assessment and management, along with the second study. So we think, our research is starting to have an impact in this area of non-alcoholic fatty liver disease. Our colleague Marinos Ilia has been very interested in undernutrition, malnutrition in the community, in care homes and in hospitals. And he and his colleagues have published a series of papers looking at the prevalence of undernutrition in these different locations in the UK setting, and looking at the efficacy of nutritional support through oral nutrition supplements, sometimes called SIP feeds, in those settings, and through the efficacy, trying to establish the cost effectiveness of this simple nutritional intervention to improve outcome in these patients. And I think this has been really very important work, showing that this nutrition support works and is very cost effective. The other really important work that he has done is producing what some people call the Southampton Report on the cost of malnutrition in England. So this is a report released in 2015, and there are really a couple of interesting figures here. So total public expenditure on healthcare in England, 2011 to 2012, was £101 billion. Public expenditure on malnutrition in healthcare was £15.2 billion. So 15% of healthcare costs are spent on managing undernutrition in our hospitals. 15%. So the burden, the financial burden of undernutrition in hospitals is enormous. Yet we have simple, effective, cheap interventions that can at least help to control this. The third thing that Marinos Ilia developed was an invention called the Malnutrition Universal Screening Tool. This is a 5-step screening tool to identify adults who are malnourished, at risk of malnutrition or obese. It's for use in hospitals, the community or care settings, and can be used by all care workers. This tool is supported by the British Dietetic Association, the Royal College of Nursing, and the Nursing Home Association. And it's widely used in hospitals and care homes in the UK and in continental Europe and more widely globally. And I think this is one of the biggest impacts that our faculty has produced in terms of translation into real clinical care of patients. The tool won the IT Innovation Award at the Health Business Awards in 2008. So it's widely recognized as being a hugely innovative, yet very simple invention. It's also mentioned in the NICE guidelines on nutrition support in adults, where they mention you have to have a validated screening tool. And they mention that the must may be used to do this. And this is the only tool that's mentioned in the NICE guidelines. So there's huge impact of this rather simple invention, I think. In Europe, the European Society for Clinical Nutrition and Metabolism, which has the acronym ESPN, is responsible for establishing guidelines and recommendations around nutritional support for hospitalized patients. And they produce proper guidelines, and they also produce many, many recommendations. And it so happens that some of us have been involved in a number of these recommendations in recent times. For example, around protein requirements for the elderly, around control of blood glucose in different patient groups, around cancer-related malnutrition and its recognition, assessment and treatment, and around the use of particular nutritional interventions in patients in the intensive care unit. So on that topic, we conducted a small but very important study of intravenous administration of omega-3 fatty acids in patients in the intensive care unit. So these were septic patients in the ICU who couldn't take any oral or enteral feeding. So they were fed continuously by intravenous feeding, which includes carbohydrate, dextrose, amino acids, trace elements, and fat. So the fat in this study was the classic widely used mixture of soybean oil or medium-chain triglycerides, or an experimental fat that also included some omega-3 fatty acids. This is the proportions of the 3 different oils. These patients received continuous administration from within 24 hours of admission to the ICU until day 6. So this was 5 days. This is a small study, but with remarkable results, I think. So one of the thinkings for using omega-3 fatty acids in this patient group is to control inflammation, because a hyperinflammatory state leads to impairment in organ function and ultimately to organ failure, including respiratory failure, but also failure of some other organs. So what we demonstrated in the study was reduced inflammation and better gas exchange at day 6 in the patients who received intravenous omega-3 fatty acids compared to control, and a much, much shorter stay in hospital. So stay in hospital was decreased by about 60% in the group receiving omega-3 fatty acids compared to control. This study has had a huge influence on the guidelines for parenteral nutrition support of patients in the ICU. But of course, having something in the guidelines doesn't mean that people necessarily follow it. So it's in the guidelines, but it's not followed so well. I think that's important. The group is also very involved in engagement, public and patient engagement and outreach, and I think Mark's going to talk a bit about that later on. But many of us are involved in many activities outside of the university, promoting awareness of our research and trying to help the public and patient groups understand how nutrition is really important to a healthy existence. We have global connections. This is changing colours. I'm not sure what the colours mean, but we have had many students from many places around the world, many visitors from many places. We collaborate very widely, particularly in continental Europe, but also in other places. And of course, we travel a lot. We're very fortunate to be able to do that. So there are many places that we're very fortunate to go to spread the message of the really excellent work being done here in Southampton. You might imagine from some of the studies I've been telling you about that we need to link with industry to do our research, but also to improve the lot for nutrition for both the general public and patient groups. So we have many, many links with industry, research funding from industry, provision of materials for our studies, of course, case studentships. We provide analytical services to some industry-funded studies done elsewhere. Some of us are consultants to industry, and we have made discoveries that have been patented and licensed to industry. So these are some of the companies that we have some sort of link with or have had over the years. So what I've told you about is our work across the life course, highlighted some of, I think, the really important things that we've done, said something about our engagement, our enterprise, and a bit about our impact, because I think our impact is extremely strong. So let's look ahead. So in the relatively recent past, we have 2 new PIs in our group, Caroline Childs and Jazz Sethi. And I think these are both really valuable additions to our group because they bring new interests, new skills, new expertise that are really going to be vital as we move forward with our research in nutrition and metabolism. So Caroline's very interested in microbiota and the impact of prebiotics, probiotics, and what we call immune fitness, enabling the immune system to act in the way it should when it meets a challenge. Jazz is an expert in the area of immunometabolism in the context of tissue remodeling and metabolic disease, particularly around adipose tissue, and she has a wealth of experience of really wonderful animal models that we can do more mechanistic work with to support our human intervention studies. Of course, we will continue to work in areas of strength, life course nutrition, obesity, fatty liver disease and diabetes, fatty acid metabolism and functionality, microbiota and so on. And I think our core is really translation from bench to bedside or from molecules to man, and of course we will stick with that. So just a picture of some of the things which are happening now. So I mentioned already non-alcoholic fatty liver disease and the interplay between the liver and adipose tissue, but the feeling now is that the gut is also involved in driving non-alcoholic fatty liver disease, and that an altered gut microbiota, what I called dysbiosis before, is somehow a driver of both altered metabolism and inflammation in adipose tissue, but also of the hepatic changes as well. And therefore the view is that correction of the gut microbiota imbalance could have an impact on the severity and progression of non-alcoholic fatty liver disease. So funded through our BRC, we have been conducting the INSIGHT study led by Christopher Byrne. This is an intervention with a combination of pre and probiotics in 100 patients with non-alcoholic fatty liver disease and a randomized controlled trial design over 1 year, looking at whether if we change the composition of the gut microbiota through this intervention, we can influence changes in liver fat, but also in cardiometabolic risk factors and in the functional aspects of the adipose tissue. So that study is almost finished recruiting, has finished recruiting and is almost finished its intervention period. So we'll have data from that from early next year on. Another interesting study that we're involved in has the acronym PRINCESS. So this is a study in 330 elderly care home residents who are randomized to control or to a combination of probiotics for a 1-year intervention. And the main outcomes are antibiotic prescription, GP visits, hospital visits. Of course, we're also or our collaborators are measuring the microbiota, and we're also measuring many different immunological parameters to look at the link between altered microbiota, altered immune function, and use of antibiotics in illness in these care home residents over the intervention period of 1 year. Then just to say something, I think at the end, about Jazz Sethi, who, as I said, is very interested in immunometabolism in the context of adipose tissue and obesity, asking questions like, how is nutrient availability coupled with adipose tissue expansion? What determines the limits on adipose tissue expansion? And how can inflammation reprogram what adipose tissue is doing, and what might be the regulators of that? So these are nice pictures from Jazz, showing the differences between lean and obese adipose tissue in mice, although actually the picture in humans is very similar to this, showing the infiltration of macrophages into adipose tissue in obesity, which is really triggering the inflammation, which has an adverse impact on insulin sensitivity and more widespread systemic effects like perhaps induction of fatty liver disease. So I think the take-home messages from my talk is that we believe We are a high output, internationally recognized group. Other people may not believe that, but we're happy in believing that. That we use the life course and translational approaches as the core to our research. We're interested in the maintenance of health and optimal function, and in both the prevention and treatment of disease through nutritional strategies. I think we have strong impact and engagement, and we have new staff creating new opportunities for us. Thank you very much for your attention.
Thank you very much indeed, Philip, for that tour de force. You'll notice that, as in the best party conferences, during the presentations, things fall off the walls. So, Philip, you've really given us, I think, a real insight into why your leading role in what was the nutrition BRC and is now the nutrition and respiratory medicine BRC works across the life course, and why nutrition in early life, in middle life, and in old age is so important. And you've really shown us how the incredible, I forget the phrase you had, high impact, high output, highly respected, international, all those things, how this is really leading now to potential interventions and indeed to monitoring tools, because of course what gets measured gets managed. The other thing that I forgot to stress earlier on, but I think you stressed it very well, is how much this is an international story. The fact that it's been calculated that malnutrition, by which we mean, of course, undernutrition and then overnutrition, and both, as Lucy was saying, in a kind of mismatched scenario, especially in low and middle income countries, is an enormous problem. And in fact, the Stockholm policy group has shown that actually malnutrition underpins progress to all the Sustainable Development Goals, all 17, you can trace back directly or indirectly to malnutrition. And you've also, I think, stressed beautifully the fact that it's not just a case of saying, oh, well, we know what we need. If people are iron deficient, give them iron. You know, if they're folate, it's more than that. You can't just chuck in some micronutrients and hope that you will change things. I'm sure you've got lots of questions, folks. I've certainly got many, but if we can hold them until for another 3/4 of an hour, we'll have time, I'm sure, just before lunch. One of the achievements, I think, when we established the Institute of Developmental Sciences was to bring together not only the group that was following David Barker's work on developmental origins of health and disease, but also the group that was extremely strong on genetics and genomics. And so it's a great pleasure now to move to that side of the story and to hear about the advances in genomics and genetics. And I think Sarah's going to present initially. Thank you.
Thank you very much. Thank you, Mark, for the introduction and the invitation to speak here today. My name is Sarah Ennis. I'm Professor of Genomics in the Faculty of Medicine. And today, to talk about human genetic and genomic medicine is myself and my colleague, Professor Diana Borelli. And we're going to do a double act to try and very rapidly cover some of the breadth of research and clinical translation studies that we've been involved in over the last decade. From across the human genetics and genomic medicine theme. So this work represents obviously our own work, but the work of the entire group at Human Genetics here in the Faculty of Medicine and Genomics, but also colleagues. We have very, very close affiliations across the road with the Princess Anne and the Wessex Clinical Genetics Service, and up in Salisbury with the Wessex Regional Genetics Service. So it's all of these people together. You'll appreciate we're trying to get lots in, so it's quite high level. And I'm going to start by talking about two of the sort of blockbuster theme areas in genetics and genomics over the last decade, which are genome-wide association studies and next-generation sequencing. So that's what I'm going to sort of set the scene with. So the genome-wide association studies were the very first studies that began to look at common disease genetics. Towards the end of the last century, we really had quite a good track record at identifying genes that underpinned single gene disorders. We'd identified many thousands of them, but we'd made comparably no success and no breakthroughs in identifying the genes underpinning common diseases or oligogenic disorders. So these are disorders where more than one gene and probably the environment play a substantial role, such as diseases that have been mentioned already today, type 2 diabetes, Alzheimer's, cardiovascular diseases, all the autoimmune diseases such as asthma, inflammatory bowel disease, psoriasis, some arthritis. So all of these things are underpinned by many genes, and it's much harder to pick up the signal to identify the underpinning genes in those diseases. And genome-wide association studies have enabled that. And I don't think anybody escaped waking up to BBC News some morning saying, scientists have found the gene for. And that's come about as a result of many of these studies. All of those were enabled first and foremost by the fact that we sequenced the human genome, and we had the first complete draft in 2003. We're still getting drafts out at the moment, but the first complete draft was back in 2003. And a couple of years later, the very first successful genome-wide association study, and the reason why every research council funding agency across the globe was inundated with applications to conduct very expensive genome-wide association studies, was on the basis of this study by Josephine Ho in Harvard. And they had a very small sample of individuals with age-related macular degeneration and some control samples without, and they managed to identify a very important disease, or a disease gene, for AMD. So that was the first successful GWAS, and it led to, up to today, we have now almost 25,000 results implicating different genes for different common diseases catalogued in the GWAS catalogue. So many of these will have very, very small effects. Some will have more modest or larger effects, but it's all these genes acting in conjunction with the environment that lead to a disease susceptibility. What's less known to this sort of big picture is the maths and the foundation upon which it was based. So it was actually our group here in Southampton, and work by Collins and Morton, actually at the end of last century, that identified that these very, very simple mutations called single nucleotide polymorphisms. So we've got 3 billion base pairs in our human genome. A mutation at just one base pair out of those 3 billions is called a single nucleotide polymorphism, and they're very abundant. Everybody here in the audience today has about 3 to 4 million of them in their own genomes. So hugely abundant markers. And identifying that they could be useful in association studies was a very important factor and a very important step in their use later on. We also developed the first linkage disequilibrium maps. So this is a terminology to denote sections of the genome that are co-inherited. So we inherit chromosomes, but chromosomes, one end of one chromosome is not always transferred with the other end of a chromosome from one generation to the next. Recombination breaks it up. So these are principally maps that denote the points across the genome where recombination happens. And we show that using SNP data, single nucleotide polymorphism data, and using them in a statistical setting, we can pinpoint the regions of the genome where recombination breaks down. And that's very important for disease gene mapping. And we were able to verify it working with Alec Jeffreys, who was observing this in the laboratory by looking at sperm genotyping and seeing where recombination was breaking up the chromosomes in real time. And we were able to reproduce the same thing looking at these LD maps. So that was very important, and it enabled the development of these arrays, these large panels of SNPs that would be powerful in genome-wide association studies. And we worked with industry to help develop the first of these SNP tagging panels that could be used for genome-wide association studies. We then used their data to help us make whole genome-wide LD maps of the entire genome for the human species, but also for sub-ethnic groups across the human species. We then used the panels ourselves locally for very, very many disease areas and very many studies. We have over 120 different publications. You'll appreciate I didn't want to put them all on this roadmap, but across many different areas, including cancer, rare disease common diseases such as asthma, inflammatory bowel disease, ophthalmology, and we've published many of those as lead and also as strategic collaborators in global meta-analyses of very, very large population groups. Of course, finding a susceptibility gene is not the end of the story. You then have to figure out, well, what does it do? How does it work? Why is it a susceptibility gene to cause asthma in one individual? Why do mutations in that gene bring about disease? Because only then can you begin to understand, well, how can we intervene? How can we change? How can we develop drugs and personalized treatment for those individuals? So obviously, and again, this is absolutely not exhaustive, but there's been a number of local studies in following up the function of these genes in trying in the next step to get it closer to translation to patient benefit. So happening concurrent with all the genome-wide association studies is the dawn of high-throughput genomics. So I think everybody is probably familiar with Fred Southern and the procedure of Sanger sequencing. It has been a gold standard and a revolutionary mainstay of both research and diagnostics towards the end of the last century and certainly the start of this century. And it is still the main form of sequencing in many places across the globe. However, there has been a shift over the last decade, and principally starting at about 2007, where there were major technological changes in the way that we sequence to enable much, much higher throughput. One of the key methodologies that's used by Illumina, which arguably is the global forerunner in producing the technology to enable this, is sequencing by synthesis, where instead of developing individual strands of DNA and just attaching a light to the end of each strand, you're actually, you've got your target DNA and you're adding on individual nucleotide bases, giving off a light, taking away the color, adding the next one, and using high-resolution photography to capture this and have millions of experiments on a tiny, tiny little wet lab the size of your thumbnail in one of these next-generation sequencing machines. So that brought about a huge step change in what we were able to do in genetics and genomics. And this is a graph that's been much shown in the genomics area, but it's a very important graph. It has over here the cost of a genome, a human genome, and you'll note this is a logarithmic scale at $100 million. So just at the turn of the century, it's $100 million to get one human genome. It's not the sort of thing you're going to buy for somebody as a Christmas present. It's a little bit expensive. But then on this logarithmic scale, as you reach along here, and a key point at about 2007 when this step change in the technology happened, the cost absolutely plummeted. And this is not the most up-to-date slide. You'll see it stops at 2015. It's now well under $1,000 per genome per individual. So from $100 million to well under 1,000 for an individual. So we do now have the ability, instead of sequencing an entire species, as we were so delighted to have the first map of the human genome species in 2003, now we are actually able to apply this technology to an individual on a case-by-case basis. And it revolutionized the way we could look and the throughput of genetic data we could gather. It is a disruptive technology. It has changed a lot of the methods, and a lot of the skill base needs to change to adapt to this new technology. But it has enabled these huge amounts of genetic sequencing, obviously not just in humans. We can do it in the microbiome, we can do it in viruses, we can do it in bacteria, we can look at DNA, we can look at RNA. The data are high quality. There are some other issues. Because the data are vast, we need to find very great supercomputers, such as the Iridis next generation that we have here at the University of Southampton, that enables us to process these very, very, very large files. But then we also need to store those data. So it brings a storage cost with the data as well. And of course, it's one thing generating a huge amount of genomic sequencing data. You then have to interpret it and bring it from bench back to bedside, back to the patients to their benefit. So interpretation and functional analysis of the variation that we find is incredibly important at the moment. We've had too many successful applications of NGS to talk about, but just as a bit of a roadmap as to what's happened, we actually had our first pipeline for the analysis of genomic data in 2011. So this was principally looking at exomes. So instead of looking at the whole genome, where we look at 3 billion base pairs, we just sequence the sections of the genome that code for protein, because it's assumed the vast majority of disease-causing variation are in those bits of the genome, and that's called the exome. So we were applying that in 2011 and generated our first bioinformatic pipelines. We were applying it to congenital anomalies. We were already looking at tumor germline pairs, where we would sequence an individual with a cancer. We would sequence their native germline DNA, but then we'd also get a biopsy from the tumor and sequence the tumor and look at those genetic changes that were specific to the tumor that could act as possible targets for drug therapy. So these were some of the very, very earliest studies in cancer genomics. And we were also beginning to apply it to some common and autoimmune diseases. By 2012, we were applying it to look at clonal evolution in cancer, how cancer changes over time and develops these subclones, some of which might be targeted by a therapy, but some, importantly, others of which might not. So using this next generation sequence to track the actual evolution of tumors. By 2013, we had successfully identified very rare variation in a common disorder. So the genome-wide association studies, which had worked so well in the noughties, had found very common variants that may underlie asthma, inflammatory bowel disease, and other genetic disorders, but they were very poor at identifying rare variation. So we were starting to use this sequencing data to identify some of the missing heritability in those common disorders. We were also identifying novel genes in lymphoma and using SNP panels to help develop methodologies in next-generation sequencing. In the years that followed, we were using it to successfully identify very low-level mosaicism in cancer, so where individuals do not have a very clear signal of the genetic mutations that underlie their cancers, and it's only a very small percentage of cells that might hold a predisposition gene for a cancer. And we were using very, very deep sequencing, generating lots of data to pick up these low-level mosaic cases in cancers. And we had myriad clinical case studies whereby patients and families with very rare diseases, which all the sequencing analysis, Sanger sequencing of genes prior to that had failed to identify the underlying genetic cause, we were now applying these technologies to successfully identify the genetic causes of diseases. And so we've demonstrated, or we're one of the groups that demonstrated the very objective power of going in and being able to sequence vast amounts of genetic sequence data and identify things perhaps where you don't already know the answer, or it's not led by a very clear clinical presentation of the patient. You can still generate these data and identify causal mutations. We had done done quite a bit in pediatric immunology, patients coming into this hospital, little children with nasty immunological disorders, and leading these to change treatment in these children. And I'll give one example of those in a minute. We'd also used the technology to identify new rare disease genes, so new disease genes underlying or single gene disorders, but also new disease genes for common disorders. And then by 2015, of course, once we've proven how powerful, along with obviously global efforts around the world have proven how powerful this was, obviously the demand was to get this into standard clinical diagnostics. So we work very closely with the NHS hospital here and help develop the clinical exome pilot and bring in the bioinformatic pipelines to start running a small pilot of 5,000 genes that we would be able to screen in individuals presenting at Wessex Clinical Genetics Service and at other parts of this hospital to routinely integrate this into the hospital here. But of course, such initiatives are also happening nationally as part of the Genomics England project, and we were successful in establishing this hospital as the headquarters for the Wessex Genome Medicine Centre, and that's now recruiting to the 100,000 Genomes Project, which is this national project to implement whole genome sequencing into the NHS. I think I have time just to show you one particular case study. It was an individual, again, who came into this hospital. It was a little boy who presented with severe combined immunodeficiency, or boy in a bubble syndrome, where simply his immune system doesn't work against common pathogens that all of us would expect to be able to deal with and handle quite healthily. So he was a recurrent patient, very often frequently ill, missing lots of school, no idea why. They had sequenced, I think, about 8 severe combined immunodeficiency disease genes and found no reason, no underlying genetic cause for his presentation. He was listed for a bone marrow transplant. A donor had not been found, and mum fell pregnant again, so it was decided to delay, wait and see if this new sibling might be a good donor. The new sibling was born, but unfortunately was diagnosed very soon after birth with the same condition. So now we had proof positive that this is definitely looking like a genetic disorder with 2 brothers inheriting the same condition. And there was no more genes to test, no more known genes to go and Sanger sequence and find out what might be wrong. So it was brought into the research setting, and we conducted whole exome sequencing on these individuals. This is a very complicated slide, and that's all it's supposed to show you. It's a very complicated slide. We had some immunology results that suggested folate was helping stabilize some of the blood markers in these boys. And this is the very nasty, intricate pathway involving folate metabolism. To go and sequence every gene involved in this pathway would cost more than the NHS can afford, to put it bluntly. It's about £1,000 a gene to Sanger sequence a gene. So it would not have been practical to go and do this. But we generated whole exome sequence data on the brothers. We, first and foremost, due to discussions with the immunologists, went and zoomed in on all the genes in this pathway, because we had the data on all genes across the genome, but we zoomed in on all the genes in this pathway and very quickly identified a critical gene, MTHFT1, which is involved in steps over here, and at position 51, where it's supposed to be a leucine amino acid, it had a proline. And these boys, both brothers, had it, as well as mum. This is a diagram of the MTHFT1 protein. This is the binding cleft where it binds folate. This is the normal leucine. And if you put a proline, which has a different shape and a big kink in it, if you pop that in here, it changes the composition of that binding cleft. And it will obviously then change the function of that protein. The boys had inherited it from their healthy mum. So that in itself was not the full story, because this is not a dominant picture of inheritance. It appears to be recessive. So we've got a smoking gun in this gene in the right pathway where they're folate sensitive, and we've got one hit from mum. We went back and interrogated the data and tried to find some second mutation in the gene that they might have inherited from dad. And they had not. And this is, we spent quite a lot of time on it and ended up finally finding the cause, which was, so this is the MTHFT1 gene across here. This is exon 13, so a very important section of this gene that codes for amino acid. This is a different individual done on the same chemistry at the same time, and we get this amount of sequencing data from them. This is one of our boys, and we only get about half the sequencing data from them. So it was by analysing not actually the mutations, but the abundance of sequencing data, we were able to determine that in actual fact, these boys each had a deletion ranging 1,700 base pairs from this section of the gene to this section of the gene, which completely removed this exon 13 section of the gene. So the boys have now got these 2 mutations. So on one copy of their chromosome, they have a mutation at position 51. And on the copy they inherited from their mum, they have this deletion of exon 13. In actual fact, we went on to prove that this version gets completely degraded, and so the boys have 50% of the protein, and that protein is not working very well. And so this was determined to be the diagnostic cause in this case. Both boys were taken off the bone marrow transplant list, which in itself is a hugely expensive procedure, not without its own risks to either the donor or recipient. And they're now being treated with folate and being given all their childhood vaccines and proceeding and back in attending school. So I think I chose this example to show what I think is one of the secrets of our success over the last 10 years, and that is our critical collaboration. Obviously, we've got many groups within the University of Southampton: basic genetics with clinical genetics, epidemiology, informatics, increasingly mathematics and high-performance computing. But with our partners on the other side, which is the hospital, which gives us priority access to patient samples and data, obviously all the clinical expertise, R&D and clinical trials. And it's on that bedrock that we can then take our research out nationally and internationally. So at that point, I'll move over to Diana.
Thank you, Sarah. So I wanted to show you how well the clinical genetics and the genetics groups work together. We have a very motivated, research-active clinical research group associated with the Comprehensive Research Network. Within that group, we have PIs for multiple local and national studies, with great strengths in cancer genetics, dysmorphology, prenatal genetics, and consent and ethics. I can't describe all their work to you, it's huge, but I thought I'd just show one example, which was our role in the Deciphering Developmental Disorders study, that's DDD, which has generated many high-impact papers. This was one of the first, and we were all very involved in. The team recruited over 700 cases to this study, and importantly, it's generated many new research projects over the 5 years of that particular study, which are ongoing and exciting. The example I've put there is a syndrome that we described called Pura syndrome, associated with the DDD study. And this has now generated many new research studies. Members of the team, well, actually here in the top right-hand photo, that is an old photo, but that was when the team was first formalised 10 years ago, and of course has grown since. Many members of this team were pivotal in establishing the Genome Medicine Centre here in Southampton. And of course our alliance with the 100,000 Genomes Project, which is incredibly successful. DDD was the forerunner of this, really. And the GMC here have recruited over 2,000 patients to the 100,000 Genomes Project, and we're now receiving our first results back, really changing how we diagnose patients. Our clinical team have been involved in, I was trying to count this yesterday, with at least 20 new genes discovered or genes associated with disease, and probably more. We were just trying to count the ones that we directly led or were involved in. We're heavily involved in precision phenotyping, these disorders, which of course is important for management, and the example I give here is the P10 syndrome. And the pyridoxine molecule there, which I'm sure you all knew was pyridoxine, was there to remind me to describe a case to you that really had high clinical impact, to demonstrate the importance of this. So this was a girl, 19 years old, that suddenly started having seizures in her teenage years. Really quite severe, so she couldn't go to university. She was sleeping a lot of the day. It was really disrupting her life. And with the clinicians, help, we undertook exome sequencing and found a mutation in the gene that causes pyridoxine-dependent encephalopathy. Now, this was thought to be a neonatal seizure disorder, so when we've expanded the phenotype, we've shown that actually this can occur later on. But also, we now realized how we could treat this girl. And with pyridoxine, her seizure stopped and she was essentially cured and could continue her normal life. And this would not have been possible, getting the diagnosis would not have been possible without the whole team coming together. So we have, in the clinical team, we have physicians and we have clinical scientists. We collaborate, of course, with our regional genetics service. And they have developed new tests, new genetic tests in hematological oncology. We also led the way in establishing the use of new technologies. For example, a new technology at the time of array CGH. We were the first in the UK to really use that. And of course now the clinical exome, which you've just heard about. Despite this team being mainly clinical, they regularly generate approximately 100 publications a year. So very research active. And together have developed this Wessex initiative in genomics medicine with many different branches. We're very involved in training and education, not only training PhDs and researchers and academics, but also with Health Education England, setting up this fully funded Masters in Genomic Medicine that is hugely popular and very successful, along with a primer in genomic medicine. We'll have our first full-time graduates in December, so that's very exciting. We undertake a lot of outreach and patient and public involvement and engagement work. These are just examples. For example, a Pint of Science, events at the Winchester Discovery Centre, and we visit a lot of schools, educate school children, and are very involved in patient groups, patient foundations. Of course, we are very aware that we can find the gene fault, but we need to know what it does and understand that very complex relationship between phenotype and genotype and how we can translate that to the bedside so that we have a continuum of improving healthcare, disease prevention, and also personalizing medicine. So we investigate the link between that gene change and the protein that it makes and the steps in between. Investigating the mechanism by which that happens is important, particularly for intervention and future gene therapies. So I have put here an RNA example using splice switching oligonucleotides. We have shown that we can decrease the breast cancer cells and that we can change the function of the protein in ataxia-telangiectasia. We have world-renowned epigenetics groups. People within that group have delineated the clinical characteristics of imprinting disorders along with the molecular mechanisms involved. We have an imprinting short stature disorder named after one of our own researchers. And apart from rare disease, there is a lot of work within the group of epigenetic mechanisms in common disease such as atopy and asthma. But ultimately, we want to personalize genomics. So within the disease groups of cancer, monogenic disease, and complex disease, we can now generate, as you've heard from Sarah, genomic data. But we do need to understand it. And a lot of our current work is in this field. So we are improving the way that we analyze the bioinformatics, the way that we interpret these sequence variants, with great input from our clinical colleagues to make new diagnoses. But we're also aware that further experimentation is needed, and we're undertaking that through different models and tissues, through RNA work, through protein work, epigenetics, with biomarkers, to improve our functional understanding of what these variants mean, without which we cannot generate rational therapeutic modalities. And without this, we want to go forwards to clinical trials, collaborations with pharma, and truly personalize medicine and undertake interventional genetics. We're currently working very hard on integrating all the omic technologies and our omics data. And this is especially important to interpret our work. So in this example, we are using next generation sequencing. This is an autoimmune example. Immunology, transcriptomics, metabolomics, the microbiome, pharmacogenomics. And all this generates a huge amount of data, and we realize that it's important to have links with our colleagues in mathematics and computer science and use new techniques there to interpret that, such as machine learning. At the same time, this all needs great clinical input and data. And putting all this together is our future, and will improve our research output doing this. We will obviously aid interpretation, we'll understand the function of these genes, and this will impact bench to bedside and clinical translation. Thank you.
Well, thank you very much, Sarah and Diana, for making an incredibly complex field so clear, and for demonstrating, I think, just the power of this amazing technology that's literally changing year on year. But I think you also alluded to some serious challenges. The fact that one has to try and not just find these genetic sites, these genomic sites, but actually translate them into function, which is a big challenge. And I think the case studies that you showed just show how actually you can do that in proof of principle and save lives, change the future for some kids or some adults, some teenagers, and that really there's an enormous future here in bringing the genetics, genomics, the epigenetics, which we'll hear about after lunch, and environmental influences all together. So, grateful to all the speakers for keeping to time. We've now got 10 minutes or so for some questions if anybody would like to rise to the challenge. We've got a roving mic here. So who would like to interrogate any of our morning presenters? What a shy audience. How surprising. No questions at all. You can have a longer lunch break if you like. Oh, Cyrus, well...
Someone's got to start. So actually, could I first start on behalf of all of us by thanking yourself, Lucy, and the team for a terrific day, and all the presenters for a great morning. So I suppose that my question is one that I'm not sure who would rightly answer, and maybe you can kick off with the approach to integrating across such a wide scientific agenda, but where you can sort of reduce the target question to a really rather simple one about the way in which very early influences, be they fixed genetic, altered genetic expression, or environmental exclusively, modify your chances of getting a disease or dysfunction that often arises quite late in the life course. There are so many fantastic vignettes, but it's the way in which we go about sort of having put this into the schema of etiology and intervention, the way in which we actually go about making the step changes and picking the right questions in the science. Because that's really what the symposium 10 years from now will need to have shown us that we've done.
Yes, thanks, Horace. I think that really is it's $64,000 doesn't buy you very much these days, does it? Or get quite a lot of sequencing by the sound of it. And of course, I'm not sure if we really know the answer. We started really from observational studies in epidemiology, didn't we? That's what brought me to Southampton and Lucy Green to Southampton, to see if we could attack that question from the other end and say, oh, well, if we try some animal models and we look for some mechanisms, do those two things, the population and the lab-based science, kind of join up. But now we're at the stage where we need to think, okay, so where do we go with that? And that really gets us into big data territory, doesn't it? And I guess it comes down to the quality of the data that we have, as well as the ability, as you say, to work across disciplines. So I think with some of the new cohort studies that we're starting, for example, the study looking at the NIPA trial, looking at interventions pre-pregnancy and then following the offspring all the way through. Of course, the Southampton Women's Survey, the pioneering study starting pre-conception. We are going to be able to assemble that data. Then I suppose with the opportunities with new NHS databases, it may be possible to probe that data and to look back, as you started to do with Hertfordshire, looking back and then joining that up with the prospective kind of approach. I'm not answering your question, but it is going to be a major challenge, I think, to bring those things together. But we probably have all the strengths here to do it. And we've got great expertise, as Diana said, across into maths and engineering and physics and chemistry, as well as the social sciences. It's possible, but a real challenge. What's your view on how we...
Well, I suppose that my reflections on just the couple of hours in which I've learned so much about what we, as a larger group, do here, suggests to me that we need to sort of create an environment in which the scientific ideas and the resources that we have are able to feed off each other, perhaps, and I don't mean this in any way to sound critical, because I'm part of all of this, but feed off each other in a more productive way even than the fantastic productivity we've gained. I mean, we're clearly internationally competitive in this, but I still think we're not using our resources in an added value way as well as we might do. And perhaps one of the thoughts that might emerge from this would be to sort of revisit this on a more regular basis than, annual or decade intervals might permit.
Thank you. Yeah, well, that's a nice cue for those of us who work here, because obviously, having got together today, the question we'll be wanting to ask you all afterwards was it a good idea? And if so, how might we take this forward in the future? And if not, how might we take this forward in the future? So that's something to be thinking about, and you've absolutely put your finger on it, Cyrus. Are there any other questions, or are you all hungry? This one is Richard, I think, up at the back there. This keeps our helpers fit.
Thanks very much. And just to echo what Cyrus said, it has been a truly engaging morning. This is really a question for Phil, Lucy, Mark, and yourselves. So this is 10 years in. We've got some very exciting science that shows the importance of nutrition, the importance of dietary intervention. There have been a whole series of very exciting clinical case studies shown here, and yet in some ways our public engagement, and I know this is coming later on, in getting that message across of, we've had lots of government initiatives about 5 a day, etc. Where is, if you like, the mismatch between the science that is so compelling and being able to get this message across stronger and engaging the public, and how do we change that within IDS, because I still think we can do that. And I just wanted to get views from the three of you on that.
Do you want to answer that, Lucy, or have a... Are you still mic'd up, or...
No.
Oh, you've lost your...
I think on a national level and from our small activities as individual groups, we've got great stuff going on. You've seen small snippets of it in terms of engaging public and people who are beneficiaries of this. But I think it obviously has to be needs to be taken to a wider stage, and there is a lot of advocacy going on to put this under the noses of people that can make those big decisions and make those big policy decisions. But that's a really hard-fought battle, which I think, Mark, probably you should pick up on.
And I'll do that, if I may, after lunch, Richard. My stomach's rumbling. Thank you very much for coming along so far. Lunch is outside. Please look at the posters, because some of our younger team members have put a lot of time into them. Please look at the trade stands, because obviously they've paid for your lunch. And of course, if you don't get your little card stamped, you're not in the running for a prize. If anyone from outside would like a tour of the IDS, then please meet at reception about 1:10, and we'll take you over there.